Perfect. Meeting is recorded. Okay, so let's get started. Hi, everybody. Happy Monday. Hope you guys had a great weekend. For those of you who are potentially on spring break, welcome back when you're watching this. But today we're going to continue our series of seminars and lectures talking about pluripotent stem cells specifically. Today we're going to be discussing or doing a little bit of a deeper dive into feeder-free human pluripotent stem cell cultures, and sort of building on what we talked about last time, where Tom mentioned a bunch of really nice base concepts that we're going to build upon together today, and we're going to hopefully even go through some videos and kind of share some really cool images and videos that we can talk about together. I should mention, just like always, please feel free to interrupt, come off mute, ask questions, drop them in the chat. The more engagement that we get, the better I think you guys will be able to absorb some of this material throughout. So please don't hesitate to speak up. But sort of as a quick review, Tom introduced this last time, right? The four big factors that we have to consider when we look at growing human pluripotent stem cells in adherent or monolayer cultures, right? We have to look at the matrix. What are these cells sitting on? Tom mentioned this, but pluripotent stem cells do not like to sit on plastic, right? And by sit, I mean they will not be attached very well to plastic. So we have to provide them with some sort of matrix. Before, we were using the inactivated mouse embryonic fibroblasts, MEFs or feeders, as some people call them, in order to have that base layer that then our PSCs can then be cultured on top of. But as we've mentioned before, there is some disadvantages to having a feeder dependent culture, primarily being the fact that you'll have two different cell types, right? Mouse embryonic fibroblasts with human pluripotent stem cells. And so we can start to replace some of those feeders with specifically with different matrices to go into what we call a feeder-free culture system, which is what we've been mainly talking about today. But aside from the matrix, right, we have the cells themselves. There's intrinsic differences between different cell lines. More importantly, potentially the media, right? What are these cells being fed, or what are they consuming in terms of nutrients? If you remember, we talked about this a little bit, but pluripotent stem cells are a transient population in vivo, which means basically that those cells develop in the early embryo and then they differentiate, right? They don't continue to reside in our bodies today. So we have to make sure that if we want to maintain them in that state, that we're providing them with very specific nutrients, and we'll talk a little bit more about that. But also the way we handle these cells, right? Passaging, cryopreservation, how we actually interact with these cells also matters because they are a sensitive group, if you will. Because of their sensitive nature, if there's a big significant disturbance that will stress them out, they tend to either differentiate or die, which again is not favorable for their maintenance, specifically. So we're going to be dissecting each of those different aspects today into a little bit more detail. But before we do, I kind of just want to briefly go over a refresher of some of the different morphological features that you have to look for in your pluripotent stem cell cultures, right? To make sure that they are healthy. Because at the end of the day, how can you tell if you're doing a good job, right? So when you're looking at your pluripotent stem cells under the microscope, there's a bunch of things that you have to pay attention to, right? The first is how close these cells are together, right? Pluripotent stem cells really like to be in very close proximity to each other. In fact, we have a term for this we call our colonies, which is how these cells grow, right? In colonies or packs. We ask how compact a particular colony as an indication of how closely attached the cells are. The second thing that you'll probably notice very quickly is that the pluripotent stem cells will have or will be in this compact colony and the colony will have refractive and defined round edges, right? So, um, the best way I can explain this is if you look, it almost looks like there's a somebody shining a light from behind of the colony, right? There's like a little glow around the edges. And that's because the edges of the colony are slightly beveled up. This is how the morphology of the cells, they're not particularly flat. And we actually want to see that, right? You want to see a nice round refractive edge up to your colony. And then the other thing is that we have to or we have to look at the size of the nucleus relative to the rest of the cell, right? So if I were to zoom in, and I think I have zoomed in here, you'll see that the nucleus in PSC culture tends to hold or tends to be a large chunk of the cell size. So you have a what we call a high nucleus to cytoplasm ratio. But if you look specifically within the nucleus, right, you'll see that PSCs tend to have very prominent nucleoli. In fact, sometimes multiple nucleoli, and that's also normal in PSC culture. Now, what happens if those conditions that we talked about, right? Those four factors, something is going on and something's messed up and these cells are transitioning out of that pluripotent phase. What do these cultures look like then, or what should we expect? You'll see that you'll start to lose some of those factors that we talked, or some of those morphological defining factors that we talked about. So that compact colony will start to lose its defined edge. You'll see here that the cells here in the center may be a little bit more compact, but as they diffuse out, they tend to be further and further away from each other, and you lose that refractive defined round edge, right? You'll have a lot of heterogeneity, and the cells will tend to be a little bit flatter compared to your PSCs as they lose that pluripotency and start differentiating. Um, and you'll note that one of the easiest way to tell is a huge difference in cell size. Differentiated cells will have varying sizes compared to pluripotent stem cells they're going to be much more compact and smaller when you're looking at them under the microscope. And this can take, the form in multiple different ways, right? It doesn't always look the same. So these are just examples of spontaneous differentiation that you see over here. These cells are going to be towards more of the center, but of a very confluent plate. And they seem to be differentiating. As you can see, they tend to be flat, larger in size, and very, very clearly different than those cells around them here. Same story right on this end, on this side here, right? If you see those cells here, these cells are a lot flatter. They're a lot bigger than those ones here. And this colony is starting to lose its rounded refractive edge, right? That edge that we talked about, that nice edge, is no longer there. This is just another example where you have a colony that is differentiating on the side, but also there seems to be a clump of cells that's growing in the middle as well. And this can happen quite often depending on how these cells divide. If a colony gets too big, even if the plate isn't confluent, meaning if you have, say, a six well plate, and there's only one or two colonies in them, you can't wait for that one colony to cover the entire six-well plate because the center of that colony will start to differentiate and have these bulbous looking like structures if that happens because the cells in the middle are also going to be dividing, but they're going to have nowhere to spread to, right? So they're going to just clump up on top of each other. And typically, when we see, these signs of differentiation in culture, this is why I have this little, um, uh, you know, um, icon or image. I wanted to show what we do when we typically clean cultures, right? Or routinely manually clean cultures. This usually involves removing the differentiated cells by scraping them with a sterile tip. And this is my very, very bad art, but this is supposed to be the end of a sterile tip, right? And what you could do is if you see those types of cells, is you could scrape them off your plate so that they're no longer attached, and then you could change the media because the mere presence of these cells next to pluripotent stem cells will get the rest of the cultures to differentiate as well, right? So we have to make sure that we remove these cells if we see them routinely. Um, and like I mentioned here, this is what you'll expect to see after you scrape these cells. So you'll see that you'll remove any cells that are attached to the surface where you actually ran the end of your tip, right? So just keep that in mind, that anything that you come into contact with, you will remove with the tip, and then you could change media so that you don't have to keep these cells attached. And aside from that, right, what you
Omar, can
need
I
to
ask
kind of,
one question?
yes, please, Tom.
What's your opinion on the so-called point of no return? Got a lot of spontaneous differentiation. Just a rough estimate, perhaps.
Very good question. I think it really depends on what your cultures look like. If you can confidently address a partially differentiated area, then I would say the cultures are good to keep. And if I go back to the examples I had here, right? If if this was the only cells that I could see, although maybe this is not a good example because this seems to be just a fully confluent plate that you have to passage anyway. But maybe, for example, let me go here. If this was another colony that was much, much further away, and I knew I can confidently remove these cells here without touching this one and keeping that on, then maybe I would keep it. But in an example where, for example, you can imagine here, this culture, these cells that look partially differentiated, also potentially even here or here, there's a lot of them. And I don't think I can confidently remove all of that without leaving any differentiated cell behind. And in those particular cases, even if I know I'm putting a plate back into the incubator with cells that are differentiated, then I would have to, toss out that particular dish or well. Does that make sense?
Absolutely,
Because
yeah.
there are parts where you could see a colony and then one end of it is differentiating. And so with your tip, you could confidently just remove that whole side and remove all of the differentiated cells in one go. But if there's a lot of them, a lot of spontaneous mutations throughout the entire well, then I would say you can't confidently remove all of those cells. And it's better to go back to maybe a earlier passage where your pluripotency wasn't compromised as much. Great question.
Can I
Actually have
give
any?
us can I give some quick comment?
Please,
Yeah,
yes.
yeah. So next week we are going to do gene editing. That is the a number of people will interest in, and please attend. And sometimes depending on the during the gene we are going to target, that cause this kind of instability. So that is the one keep in mind. And another aspect is during the reprogramming, depending on the donor genetic background or mutation they have. And each donor, some line is very unstable than the other line. And then also among the multiple independent clone, their characteristic is also very variable. So in this case, what is the good choice? Just pick the stable clone, then unstable one. Yeah, that is the comment.
True. Absolutely. And you're absolutely right, Kitai. The even clones from the same donor, right? The same patient, potentially same genetic background, can give you slightly different kinetics. One can be stable, can differentiate a lot more quickly or spontaneously. So keeping an eye on them is important. And Ada is asking in the chat whether the plate cleanup should be only before passaging. So you don't have to do it only before passaging, but I would say once you do it, you have to replace the media so that you can remove those free floating dead cells that you've scraped off. and then replace the media so that the cells will have new, fresh media to make sure that they maintain their pluripotency. Okay, so for PSCs, right, typically to monitor the differentiation, if there's any spontaneous differentiation, we do daily inspections, right? We just look at these cells, make sure that the cultures are okay. If they're not, if there is any differentiation, we then go ahead and remove, right, manually clean up if we need to. And traditionally, right, we'll talk a little bit more about this, but we had to feed ourselves every single day, right? And we'll talk more about this. More modern media, you can skip feeding every other day. But typically with pluripotent stem cells, you're either feeding your cells every single day, weekends included, or every other day, depending on the media that you're using. Now, we are mainly going to be talking about, like I said, feeder-free systems, but if you are using feeders, you have to prepare those one day ahead or a couple of days ahead of your passage. So before your plate gets confluent and you have to passage it, you have to make sure that you have prepared MEFs for the next passage over. But this and this could hold definitely true for feeder free with the matrix. You have to coat with the particular matrix, but because the coating time and the preparation time is much shorter, you could do that on the same day as well. We'll talk about that in a little bit, so don't worry too much. But usually, right, so we do the daily inspections, remove the differentiation if we need to. We feed every single day. And then if we think that the colonies are and the cells are getting close to 70 to 80% confluent, this is when we typically passage our cells, right? So we enzymatically digest them or sorry, not enzymatically. We digest them into clumps and then passage them over into a subsequent plate. So again, let's talk about the individual components of our culture system here, right? So the matrix. We talked a little bit about the mouse embryonic fibroblasts, so I won't mention them too much. But other matrices that you can commonly see that can be used to coat your plastics with, so that these cells can attach without the presence of the MEFs, include laminins, right? So laminin 521 and laminin 511 are two very common laminins used. Vitronectin is another protein or matrix that can be applied here, right? So specifically, and we'll talk more about this in a little bit more detail now, but the N-terminal truncated version of vitronectin. And then another thing you can use is basement membrane extracts like Geltrex or Matrigel can be used to coat the plates in order to prepare them for PSC attachment. And Tom and I put just a little bit of considerations here for the different matrices that you may be using. Typically, Matrigel or Geltrex is the most common way that you'll see folks coat their plastics with. It's not a xeno-free method, but it is cost-effective and very commonly used. Vitronectin is another way in which you can coat these plates, right, before you plate your PSCs. It is quite on the cheaper side, relatively speaking, and it is xeno-free. But some will argue that attachment in Geltrex or a Matrigel or laminins can be a little bit better than that on vitronectin. And then finally, laminin is usually the most suitable substrate for PSCs to attach to, meaning that they will prefer a laminin substrate and they will attach really, really well to it. We'll talk about it potentially next week during the gene editing experiment, but when we challenge these cells or when we put them through a very stressful process, we tend to plate them on laminin-coated plates because they support their attachment a little bit better. That being said, laminin is usually a little bit more costly compared to the other alternatives too. And for each of those different matrices that you can use, right, there's different considerations that you have to take into account, right? Whether you need a xeno-free or a defined matrix. vitronectin and laminin are, but Geltrex is a basement membrane extract, right? So it has a ton of different proteins in there. Laminins, collagens, integrins, fibronectins, right? We talked a little bit about this last time. What is the thaw temperature that you have to maintain when you're working with each of those different matrices, right? Just remember, for example, the basement membrane extracts, Geltrex, Matrigel, these tend to polymerize if they get too warm, right? So you have to really make sure that you're handling them at the recommended storage or working temperatures. Also, the recommended dilutions will be different for each of the different matrices. For vitronectin, we tend to recommend around 0.5 microgram per centimeter square. Same thing for laminin. And for the basement membrane extracts, right, Geltrex, Matrigel, you'll see anywhere between 1 to 50 to 1 to 100 dilution as well. But finally, do not forget too that most of these uh, uh, matrices come in concentrated formats or powder formats. So you have to resuspend them in a particular dilutant. And there are considerations for the dilutants themselves too, right? So we tend to use DPBS without any magnesium or calcium, which you'll commonly see denoted as minus minus. So when you see that minus minus, that just means no calcium, no magnesium. And we tend to use, again, DPBS with minus minus without for diluting vitronectin, but DPBS plus plus for diluting laminin, for example, right? Small difference can make or small change can make a world of difference here of how your cells attach depending on the dilutant that you're using. And so we talked about matrix. I think Tom covered the kind of the cells portion pretty well last time in talking about these cells and giving you an introduction to them. But I wanted to spend a little bit of time on media as well, because I think this is a very, very important consideration. And so over the last 25 years or so now, our field has gone through a huge incremental set of changes that has allowed us to improve upon the way we culture these cells with regards to the media specifically, right? So what you see here is just a little bit of a timeline of some of the important points across the last 20 years or so of how we used to culture human pluripotent stem cells, where in the late 90s we were using feeders and then media supplemented with FBS to grow our human PSCs, right? And then shortly afterwards, we realized that FBS, the use of FBS, is associated with a lot of variability. That was very, very hard to deal with at the time. So instead of using FBS supplemented media, we actually replaced the FBS with what we call knockout serum replacement. So this is just a serum replacement that's a little bit more defined that can help reduce that variability and was actually introduced and made a good effort there to help reduce that FBS variability that we saw. But the next big jump came around 2006, 2007, where a group in Wisconsin, a very prominent group, it was Jamie Thompson's group in Wisconsin. They published a paper around the use of a defined feeder-free media system, right? Where they outlined a media that they called M-teaser with the use of the basement membrane extract, Matrigel, in order to culture these cells without the feeders, right, for the first time, right? And so since then, that same group, or I should mention that same group, several years later, also, introduced another media system called Essential 8, right? And so Essential 8, as the name suggests, and I think Tom mentioned this before, is a media that you can use to culture your stem cells with, but that only has 8 components, right? It's the bare minimum that the essential 8 components that these cells need to maintain that pluripotent state, right? And Jamie's group also used or introduced the use of vitronectin in this case as an accompanying coding matrix. And some of you may be thinking, well, why would the same group be developing two different medias, right? Well, mTeaser was really good at keeping these cells happy, but mTeaser had a lot of components as well into the formulation. So Jamie's group wanted to pare this down to figure out what would be, the bare minimum that these cells need in order to maintain that pluripotent state. Additionally, to help reduce variability you know um, from making one media set to the other, what would be the 8 components that we have to pay attention to, right? Every time we make media, because those would be the core components that we have to work around. And so in doing so, we really learned a lot about what these cells need and what components that have to be present in our media for these cells to proliferate well and then stay pluripotent when doing so. Um, a quick shameless plug at Thermo Fisher, we also developed two different medias. One media that was a slight variation on the essential 8. One of those essential components that we'll talk about in a minute, that is needed for all PSC culture is FGF2, right? And FGF2 is not a heat-stable protein, meaning that it breaks down at 37 degrees, which is where we know the temperature we usually culture cells at in the incubator. So we figured out a way at Thermo Fisher on how to stabilize the activity of FGF2, which allows you to do a whole bunch of different things that we'll talk about in a second. And then in 2017, our R&D team also did a design of experiment approach where they built a brand new media system that we launched in 2017 called StemFlex. StemFlex was specifically designed to help with modern day applications that are a little bit more challenging with PSCs, things like gene editing, single cell sorting, clonal expansion, that we're going to talk about slightly today, but also during our gene editing lecture next week too. So let me take you through a little bit more detail on some of those media systems that we talked about. Okay. The first one I mentioned was KSR, right? Replacing FBS, where the use of either FBS or KSR you'll most commonly see right now with feeder-dependent systems and not feeder-free systems. And typically, the media formulations is as such. You have your DMEM F12, and then you supplement it with 10 to 20% KSR with non-essential amino acids, mercaptoethanol, and then FGF2. And this is what you will use in order to culture your cells in, right? This concoction. Now, typically with that change from FBS to KSR, what we did see in the field is slightly better morphology of these cells as you transition due to that reduced variability, right? So just keep that in mind that even though FBS and KSR both can be used, that most people will gravitate towards the use of KSR because there's a little bit of work in the literature to help describe how KSR can be used to better derive mouse embryonic stem cell models too. And then I mentioned, right, the next big jump in terms of the media that replaces the need for feeders or the use of FBS or KSR was jumping to those feeder-free systems, right? You can see this here at the bottom. This is a nice PSC colony that's grown in essential 8 media. But notice how there's no mouse embryonic fibroblasts right on the side. In fact, the plate at the bottom was coated in this case with vitronectin before plating these human pluripotent stem cells on top. And a lot of this feeder-free culture was pioneered, like I mentioned, at a lab in Wisconsin, University of Wisconsin, specifically Jamie Thompson's lab, where he helped develop mTeaser and Essential 8 media as well, right? And now you could buy mTeaser and Essential 8 media at two different vendors, us being one of the vendors that sells Essential 8. But just as a quick reference, there is benefits to both as well. I'll describe specifically what Jamie's group also did with regards to the matrix, right? With the development of the Essential 8 media, at the time, the goal was to figure out, right, how to have a defined media system where I don't need to use Matrigel or Geltrex that may be variable from one lot to the next, or a media that is that potentially can use albumin as well, like mTeaser. So in a fully defined feeder-free system that uses Essential 8, we had to find another matrix, or Jamie had to find another matrix that could pair really, really well. And maintain the system as completely defined, meaning you know exactly what you're coding with and what the cells are actually receiving. And so, in order to do this, vitronectin was one of those candidates. So, Jamie's group actually tested multiple different variants of vitronectin, right? Where they coated the plastic with either wild type vitronectin where the protein was truncated on the C terminus. Another variant where the vitronectin protein was truncated on the N terminus. And then a fourth variant where they've truncated both the N and the C terminus as well. And what basically they found out after, a lot of work is the Essential 8 media, along with the variant of the N-truncated vitronectin, so this one right here, tend to can represent or can together be used as a serum-free, xeno-free system for culturing human pluripotent stem cells. Now, this was important because this was sort of the first time where we could show that we don't need animal origin components particularly in order to culture these cells, which gave us the hope that we can then, continue to push these kinds of applications into the clinic, where this is, the eventual goal, like we talked about, is to use these cells as some sort of repair kit in some sort of therapy capacity, right? Excellent. Now, those two media systems were really revolutionary, I have to say. They really, really changed the game because for the longest time we had a lot of trouble culturing these cells. They would spontaneously differentiate at whim, almost it seemed like, right? So there was a lot of variability in those early days, and mTeaser 1, Essential 8 really did standardize the game quite a bit. But one thing that both media components include, which is part of those 8 essential factors, is, as I mentioned before, FGF, right? And so FGF is one of those components that controls the maintenance of pluripotency and proliferation of these cells in culture. And some of you may know this as well, but if we look at protein half-life at 37 degrees for FGF, you'll notice that even in the absence of cells, if we're just putting the media at 37 degrees, right, the concentration of FGF in Essential 8 or mTeaser 1 tends to drop quite quickly, right? And this is important because this is one of those factors that is important for maintenance of pluripotency, right? So if it drops too much, then you're worried about these cells differentiating. And you can see this drastic drop within the first 24 hours. And this was the primary reason why we needed to feed these cells every single day. We needed to go in and prepare new media and change it so that we can replenish, right, the levels of FGF after they've degraded at 37 degrees. That was one of the main reasons why we had to change media, right? And uh, 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 although, that does work, but that could introduce somewhat a bit of variability into culture systems where, throughout the day, the cells are seeing increasingly lower and lower levels of FGF up until you do end up replenishing them, right? And so at Thermo, I mentioned we did develop a system by which initially in essential 8 flex, but also used right now in stem flex, where we can maintain the FGF, the level of FGF2 activity quite stable across an extended period of time, right? So this allows for two things. One, it reduces these huge dips in concentrations of this very important critical component that these cells are experiencing. But also, this allows us to then start employing those flexible feeding schedules, right? Because the FGF2 concentration isn't dropping down too drastically in the first 24 hours. I don't have to feed my cells every single day was, another potential benefit that we could use from this, right? And so we ended up employing a flexible feeding schedule with media systems that did employ this stable the stability of FGF2 activity or that approach specifically, where we can passage our cells and feed them the day after passaging, but then keep them growing in the incubator, right? Not disturb them, let them continue to grow, and potentially even reduce the handling time that can introduce a contamination. And then when these cells are ready, passage them, potentially even, allowing you to skip 2 days of feeding if you double feed the day previous. So you really have a lot of flexibility in this kind of way to follow your cells and sort of treat them as their growth kinetic really dictate, right? And when using these flexible feeding schedules, one thing you have to first look at is the maintenance of pluripotency. Are we still maintaining these cells as pluripotent? And the answer is yes, right? So in these newer systems like StemFlex or Essential 8 Flex, we can maintain pluripotency of the cells, even with the flexible feeding schedules. But if you were to do it with older media systems that have that drastic drop in FGF2 activity, like MTZR1 or Essential 8, then you could potentially start to lose pluripotency in those cases. And with StemFlex specifically, right, the goal is to continue to culture these cells onwards, right? And so I just wanted to include this slide as a reminder that even though you can continue to culture your cells in robust media systems like StemFlex for an extended period of time, right? Because these cells can grow, and as they get to that, 70% confluency, then you could dissociate them and then replate them back and continue to propagate them and passage them onwards, right? But as you passage them, every time you passage them, there is a chance that these cells might change, mutate, and or differentiate, right? And so the media systems do have the ability to skew that a little bit. Choosing the right media systems for prolonged culture is important, but also very, very important is that if you're going to go to these very high passages, that you characterize your cells routinely, right? In this case, these are long-term StemFlex cultures where we had to make sure that even at passage 60, which is honestly quite high, that the morphology of our cultures is normal, that the karyotype of our culture is normal, and that we see expression of the right pluripotency markers at those high passage numbers. And so even for different media systems, you have to really do your due diligence during that prolonged culture period to make sure that your cells are truly pluripotent and karyotypically normal. And today there are a whole bunch of different media options, and I don't want to kind of get into all the different reasons why you would use one media system over the other. Tom and I obviously work for Thermo Fisher, so I'll make it very clear we are very biased, right, as to what media systems we're using. But at Thermo Fisher, we do offer several different media systems that we've talked about, right? So our KSR supplement, as well as the Essential 8 and the Essential 8 Flex media, and our StemFlex media are good options that we can offer. But the reason why I put this slide up is more so to talk about choosing the right media that works for you, right? So when you do that, right, looking at a bunch of different parameters is really important. You have to assess whether you want to be in a feeder-dependent system or not, whether you want to work in a xeno-free system or not, the workflow complexity that you're comfortable with, whether you would like to routinely clean up your cultures to make sure that you have tabs on them, or whether you want a media systems that is a little bit more robust that can potentially take care of that for you. What downstream applications are you intending, right? Gene editing, for example, is one such example where not every media system can work as well as the other in those particular workflows, right? So choosing those or keeping those options or those categories in mind will help you pick the right media system for you. And again, as a shameless plug here, I will say for StemFlex medium, this is what we use at Thermo Fisher. We do see really nice support for our PSC cultures in a feeder-free format where we can expand these cells pretty well. And where the performance of the media specifically shines is around using our media to develop gene editing and single cell sorting workflows, where we can introduce edits into our cells and have them recover in a StemFlex media really well, up to two times faster than some of those older media systems. And even when we clonally expand, meaning after our gene edit, when we want to specifically look for individual clones, we see up to a five-fold improvement in our single cell passaging workflows, even without ROCK inhibitors. Regardless, right? Now we talked a little bit about media, matrix, what you need to think about when culturing these cells, aside from how they look like. But as you continue to culture them, Tom mentioned this, that we have to consider that these cells proliferate, right? They divide and they're going to start increasing in terms of cell number, and they're going to take up more space, right? i.e. their confluency will increase. And so when they get to a particular confluency at around 75 to 80%, that's when most people recommend passaging those cultures over into a subsequent plate or an extra plate. So we're going to talk a little bit about passaging those cells, which is part of the four different factors that we were originally discussing, right? Now, when you break up these colonies as they're growing, right? So imagine this is a PSC colony, right? As you want to passage them, the main goal here is to try to break this colony up so that you can take these cells and transfer them over into a subsequent plate, right? Where you can take cells that have fully occupied your 6-well plate, there's no more space to go. And now you can take those cells and potentially divide them into two groups, now into two new plates, right? And that's how we usually passage and propagate cells. So in order to take care of PSC colony, there's multiple different ways in which you can do this, right? You can use, and Tom mentioned this or alluded to this before, you can use different enzymes or dissociation protocols in order to break your PSC colony up, either into larger aggregates with mechanical dissociation or potentially EDTA, or you can use things like triple and acutase or trypsin to dissociate them into single cells. Now, the approach that you take really does matter, right? And I am colorblind, so maybe this isn't the best slide to use here, but I think there should be a difference in the color of the cells that are designed or designated as alive or dead, right? And typically, when you dissociate your PSC colonies into very small groups of cells or single cells, they don't like that very much. That stretches them out too much, and they end up dying in the process. So when we do our routine passaging of our colonies, that's why we recommend a more gentle dissociation approach, right? Where we can either use mechanical force or EDTA based dissociation reagents to derive cell clumps, where these cells will still be in close contact with each other, which will help them survive into the next subsequent passage. Does that make sense? I hope that wasn't too wordy. But if it is, let me know, and I can explain this again. And a little bit last time I showed you part of that video, but I'm part of this video, but I wanted to run through it together, where you can see the effect of the different dissociation reagents on the colonies themselves, right? Here, looking at three different dissociation reagents of varying strength. Versine is our version of that EDTA dissociation that we can use for clump passaging, right? This is a chelating agent that promotes the breaking of cell junctions. So you can make these smaller clumps that you can then plate over onto a newly coated plate, right? And then 24 hours after passaging, you'll notice that you get that nice morphology, that compacted colony morphology that we're talking about, right? Now, if I were to use our Stempro acutase, which is slightly stronger, right? The Stempro acutase can actually dissociate our PSC colonies into almost to a 1 to 2 cell cluster, depending on how long you keep the cells incubated in there as well. So you'll see that once we get to that 1 to 2 cell cluster, and I can package them over, they're smaller clumps that we see down. And as a result, you'll notice that once the cells sit down 24 hours later, their morphology is slightly a little bit less rounded than that of the EDTA passage cells, right? Because they're a little bit more stressed out. There was a little bit more cell death during this particular process. Now, finally, you'll see that the stronger reagent that we're showing here is our triple select, which we use when we want to do single cell dissociations. So in this case, we're dissociating the PSC colonies into single cells, right? That you could individually see them right here. And when you passage over those single cells and have them attached to the subsequent plate, you'll notice that they do look a lot more stressed out, right? And after 24 hours, which is right now. They're not as compact. They're a little bit more stretched out, perhaps a little bit more flatter, right? Now, these cells were passaged as single cells into StemFlex medium. So, because this StemFlex is particularly supportive to the cells, they can recompact after a couple of days. But you have definitely noticed, right, the change in morphology. So the way in which you dissociate your cells is super important to their health as well, and how they maintain their morphology too. Any questions about that so far? Let's see if we have some in there. I know I saw the chat go off a little bit with differentiated cells. I think we covered that.
Hi, Omar. Can I ask one question?
Yes, please.
Yeah, yeah. So just now you explained about triple select. So
Mhm.
can you give some information about triple E express
It,
and
the two, yeah,
yes,
the two are very similar. It's just basically the way in which they're manufactured, really. That's a distinction. But we typically use the select for PSC applications.
okay,
But
great.
they're pretty much the exact same thing.
Okay, great. Thanks.
Yeah. Um, okay. So I mentioned, right, that the dissociation reagent that you're using is super, super important, right? So now we have to consider that when you're dissociating your original PSC colony, it's really the size of the clumps that's going to dictate that, right? Gentle dissociation reagents like Versine can create these really good sized clumps. But if you over digest, then you're gonna create colonies that are way, way too small that can then potentially start to get too stressed out and then differentiate when you passage them. Now, at passaging, right, you can either passage your cells into a subsequent plate and split them, or you could also potentially cryopreserve them, right? So I won't go through this whole video particularly, but I wanted to just give it to you as a resource where we do have, recommendations and specifically even just how-to videos on how you can perform some of these tasks or some of these parts of the workflow, right? We specifically call out, the reagents that we're using, the different methodology that we're using that we're talking about, and specifically trying to call out any suggestions or helpful tricks to help streamline the way in which you handle these cells better, right? So if you have some time, please take a look at some of those resources that we're going to share at the end. It has some really, really good, again, just visual representations of how to perform these different tasks associated with PSC culture. Excellent. So any questions about 2D culture before I jump in and wrap us off with the 3D portion?
Hey, Omar, really quickly, this is Tyler.
Yeah.
Just wanted to ask the duration of each of those time-lapse videos you showed.
Mm-hmm.
Like,
So for typically, depending on the reagent that you're using, with Versine at 37 degrees, it usually takes anywhere between 3 to 5 minutes, depending on the cell line. For Acutase and triple, it might be slightly a little bit faster. So maybe 3 to 4 minutes is where I tend to keep an eye on them. But honestly, Tyler, I think the best way to do it is to set your timer for around 3 to 4 minutes and make sure that you're looking at your cells before and after you add your dissociation reagent. This way, you'll be able to tell, at what point do you start to see those good sized clumps that then you can carry over into your subsequent passage.
perfect, thanks.
Of course. And as part of the resources that we'll share too, Tyler, we have very, very specific, step-by-step protocols that we can share, right? Where you'll know that with, for example, Versine, you'll leave it, 4 to 5 minutes. For EDTA, it could be 3 to 4 minutes, right? All of that we can share with you for sure.
Yeah, absolutely. Appreciate it.
Of course. Excellent.
I have a question. I wanted to ask, when we culture on StemFlex, even to some cells that have been reprogrammed and expanded on StemFlex, when we get them, we expand. Most of the colonies look great. Occasionally, you'll see colonies at the edges kind of start beveling up.
Mm-hmm.
And then with time, they kind of form into a little embryoid body type thing, and then they're removed every time you change the media. I don't see that type of push differentiation in any other stem cell formulation. Is there something unique to StemFlex that causes maybe cells that were slightly on the edge to tip and then differentiate quickly, and then everything else remains is fine? Or what's causing that?
Not particularly, but I would imagine it's the growth kinetics, Angel, just because the cells tend to grow really, really well in StemFlex, right? So they proliferate really well. They're quite happy. So especially around the edges where they can start to crowd, you'll start to see a differentiation, a more readily differentiation phenotype because of how fast these cells are differentiating compared to, other more linear media systems, for example, like Essential 8. So, Have you seen this with one particular line or several lines at this point?
No, about a dozen different lines. Essentially,
Oh,
it's somewhat
nice. Okay.
agnostic. And then these colonies aren't unusually large. This is happening both in smaller colonies and in medium-sized colonies.
Hmm.
It's not in all of them. It's a small percentage. And when we use other media formulations, we tend to see more spread out fibroblast like differentiation. We don't see that fibroblast like differentiation in these cells, right? The small percent that do show signs of differentiation are showing almost like an EB like
Really?
formation.
Yeah, I think it has to do with the growth kinetics in those lines. So what I would recommend there is just a potentially a higher split ratio. So you're able to keep these cells like a little bit further away from that differentiation potential due to the crowding around the edges specifically, right?
This isn't even with that because the recovery we're seeing with StemFlex isn't as good. Even in your own data, you show that StemFlex with uh, vitronectin show about 50% recovery compared to Matrigel. So we already split at a higher ratio, maybe a 1-to-4 ratio with StemFlex compared to 1-to-6 with other formulations.
Oh, I see what you mean. Okay. Yeah, and like I said, it really depends on what those applications are and what your particular cell lines are, right? So if they're growing really really well in those other medias, then potentially they could be adapted, to better to those other medias too, right? So there's a lot of nuanced things that we don't understand specifically about the cell lines that I wouldn't be able to tell you unless I could have a little bit more specifics on those cell lines specifically.
Uh, the New York Stem Cell Foundation lines, and then other ones we generated, and other ones we
Mhm.
got from both Thermo and other organizations. So, but we can talk later.
Yeah,
Thank you.
yeah, I was gonna say if you want to talk offline, we could definitely discuss this further. I'm really interested to see what those phenotypes that you're describing look like for sure. Of
Thank you.
course. Awesome. So everything we've been talking about has been around culturing these cells with in 2D, right? In on a matrix where they get to attach to plastic. But there's newer ways in which you can actually start to culture these cells completely in 3D as well, where you can start to utilize a bunch of different really really cool kind of technologies and factors that really, really make the use of these cells in 3D models really, really interesting because we can start to differentiate into organoids. We can start to think about the 3D biology of how these cells differentiate and potentially develop, which is really, really cool, right? And specifically at Thermo, we do have a particular media that we're going to be mainly discussing because that's where our main experience is, called stem scale PSC suspension media. Um, but before I get into that, typically, suspension culture, as we're calling it, or 3D culture of pluripotent stem cells specifically, is not like a brand new concept, right? There has been a lot of really, really cool work has been done on how we can potentially start to move into more 3D or suspension-based system beforehand, where previously there was multiple different systems that can be employed, albeit with their challenges as well, right? So initially, we would take these pluripotent stem cells and then we would coat a microcarrier with some sort of matrix, and then have these cells attach to these microcarriers where they can be, they can resemble a 3D structure with a little bit of curve, all right? And these microcarrier cultures, showed pretty relatively consistent cell growth, right? So like cells would grow pretty well in them. But those cultures were a little bit time consuming, hard to prepare. They're also a little bit challenging when it comes to passaging or cell attachment. I tried to depict that here where you can see, for example, some microcarriers would be fully covered with cells. Some would have some attachment, some would have none whatsoever. So it's hard to standardize that in that particular sense. And their passaging and handling was a little bit trickier. Now, A relatively easier spheroid culture to maintain was self-aggregating spheroids, right? Where we don't use a microcarrier or a matrix. We just have these cells kind of clumped together and culture them as a free-floating spheroid. And those were, again, like I said, relatively easier to maintain, but they did also there's also some tricks around the size of these spheroids and how big they can grow. And that's a very, very important consideration in 3D biology, right? On how different nutrient gradients can access the center versus the periphery of that particular spheroid, particularly with our cells, because they're pluripotent stem cells. This is very, very important, right? Because you have to make sure that these cells aren't getting different gradients across that could potentially incur a different phenotypic change, where for example, the center of the aggregate that has less oxygen, maybe less nutrients can penetrate as it gets really, really big, can potentially start to differentiate, for example, or become extremely hypoxic. And and so the goal really is to have, especially again with in this case for us with stem scale media, was to expand these cells in suspension, but also make sure that the workflow is simple, that we can do this across different formats, and that we can have a control of that spheroid size that we're building, right? And this becomes very, very important as we're going to talk more about this, but towards the end of our series. But as we get into those more clinical applications, right, of how do we get these cells into some sort of therapy? Whether I know UCLA has been driving a lot of really, really great innovations there. The mission of CERM is specifically that, right? It's the California Institute for Regenerative Medicine. It's to use these cells in some sort of regenerative capacity. But in order to do that, we have to have a huge number of them, right? We have to have on the orders of billions of cells at a time. And if we were to do that in adherent 2D culture, we can do it, but it's going to cost a lot, and it's going to take a little bit of time, as you can see at the bottom here. If we were to generate 4.5 billion cells, which I think, Tom, you might have showed this before, but this is the number of cells, of pluripotent stem cells that you need to treat approximately 1 to 2 patients with cardiomyocyte cell therapy. If you were to expand them in 2D, right, you're going to need a certain number of days, and you have to go through a bunch of different formats. With suspension-based culture, you can do it in a fewer time, potentially even saving a little bit of money or cost along the way, compared to those adherent systems, which allows us to go into the clinic a little bit easier, right? Why am I telling you all of this? What's kind of the real advantage here? Well, again, aside from particular cell number, we can get a lot of cells, but we can also start to think about doing all of this in 3D, right? Having a physiologically relevant model to study our own bodies. And the way in which we embark upon this, or sort of figure out how to get these cultures going, is we can take our 2D cultures and dissociate them into single cells and replate them into a non-TC treated plate. So these are plates that didn't have any treatment for the attachment of the cells, and no matrix either, and so no matrix coating, in a media that's specifically designed for 3D culture. In this case, we're using the stem scale media, right? And so when you plate these single cells in those non-TC treated plates, what you have to do afterwards is you have to provide some form of agitation for them, right? You have to get these cells to rock back and forth so that they can find each other, and then clump or nucleate into a spheroid that can then continue to grow onwards, right? And I actually have a video to show you that process. And we're going to talk about what we need here. Again, this is a little bit to be completely fair, and this is a little bit biased to Thermo Fisher's offering, where there are other offerings out there, but we have experience with our media systems, and we believe we can do this process really, really well. So I'll pause right there just to kind of give you an introduction. These are the different things that you would need in order to initiate those 3D cultures, right? So you need your a dissociation reagent, right? In this case, a Stempro Accutase. You're going to need a wash buffer, right? Our DPBS minus minus, so no magnesium, no calcium. You're going to need your PSC media that's designed for 3D culture, right? In our case, our stem scale media with basal media, and then it's stem scale basal supplement that are going to be added together. And then we're going to need a ROCK inhibitor. In this case, a Y27632 compound is used, and I'll specifically mention why we're using this particular compound, or why we're using a ROCK inhibitor here. And then some plastics, right, that are not treated for monolayer culture or adherent culture. And so first thing you want to do is you want to make sure that you're putting on your safety goggles or your safety gear, your PPE, and making sure that your cell surface is clean, right? Washing with ethanol. I'll fast forward, and then the first thing that I would do here is I would prepare the plates that I'm going to be seeding into. So you'll see I am adding that 3D media, so the stem scale media, into my plate and my flask, and prepping them so that they equilibrate in the incubator at 37 degrees. I still haven't added any cells yet. This is just to prepare for the cultures, right? And I already kind of discussed this. So these are the different components that we're going to need in order to start our cultures, right? So now we start by taking our 2D cells, right? So these are adherent systems. We're going to aspirate the media, and then we're going to wash with our DPBS. Notice how when I'm first washing the cells or I'm adding media on them, I do a couple of drops first before I completely dispense any liquid. And that's usually to reduce osmotic pressure on these cells, right? So after I wash my cells, I'm going to add my dissociation reagent, right? In this case, I'm adding 1 ml of the Stempro Accutase so I can dissociate these cells into a 1 to 2 cell cluster, right? And then I'm going to incubate the cells at 37 degrees for about 4 to 5 minutes. So after I do that, the cells would have lifted off, right? And I'm going to collect my cells. You can actually kind of see that the liquid in my pipette is cloudy, right? Indicating the presence of these cells. And in order to stop any further dissociation reaction, I'm going to take those cells that are in the Stempro Accutase and dilute them in more media so I can stop any further dissociation. After which I'm going to spin them down and then resuspend that pellet in fresh media with the Y27632 ROCK inhibitor added in there. Now, I'll pause this right here. Actually, I'll keep this going. So the next thing that you'll see me do here is that I'm just counting the cells, and then I'm seeding the correct concentrations of the cells in the flasks and dishes that I prepared beforehand, right? Those were the first ones that I put. And then I put them back in. Now we have the media, the cells, and we put them on the orbital shaker to allow these single cells now to start to nucleate into spheroids, right? One thing that's very, very important about these 3D systems that require this agitation or this rocking is the speed at which you rock at, right? The speed of this system is correlated to how fast your shaker is going to turn. And if the speeds are too high, the cells are not going to nucleate. And if they're too low, the cells are all going to just clump up into one very large giant ball that's going to differentiate and die, right? Any questions so far? This is just a video of the initiation of those cultures, but again, we have that we're going to send out in the resources We actually have videos for how to feed those cultures, how to passage them as well, right? And typically, right, with those cultures, what we do is after we initially initiate them, I'm just trying to find my cursor over here. There we go. So after we initiate those cultures, right, we start to see those single cells come together and form spheroids as early as 24 hours later, right? This is where we feed those cultures, typically by a 50% medium replacement. And similarly to what we talked about before with the FGF2 stability, this particular media employs that same strategy or same technology and innovation. So you don't have to feed every single day. You can skip feeding and employ that flexible feeding schedule. And when you do feed those cultures, right, you saw that I talked about a 50% medium replacement strategy. The reason why we do a 50% medium replacement is that it gives us a chance to remove spent media that, may contain factors that if kept in or metabolic waste that if kept in the culture might, cause these cells to be stressed out and eventually differentiate. But also is to add fresh new media all at the same time as maintaining the same volume, right? And the way we do this with plates is that we simply, remove the plate off of the orbital shaker, and then we tilt it at a 45-degree angle, allowing the spheroids to settle to the bottom of that plate in that 5 minutes that we wait. After that, we just simply, from the top of that liquid interface, remove 50% of the media and add fresh media, and then put that plate back onto the orbital shaker in the incubator. If you're working with flasks, this is even easier, right? There's no tilting or anything needed. You just remove your flask off of the orbital shaker. You wait for 5 minutes so that the spheroids now settle to the bottom, and then you just aspirate 50% of the media that's spent, and you replace that with fresh media that you've prepared before you again put the shaker or put the flask back onto the shaker in the incubator. And so you'll see that the important things here is that your processes should be simple. In this case, our process is amenable to automation, which is a definite plus. But two very important things that you have to kind of keep into account when thinking about suspension-based culture or 3D culture is the maintenance of volume and shear force, right? So these cells, as they're being rocked and constantly in culture, they're experiencing a lot of forces from the fluids that are moving around that are swirling around in the dish, right? If I were to take a little dish and just swirl it around, you'll see the media moving around, the cells moving around as well. So the cells inside are going to be experiencing forces, and those forces are dependent on how much media is there, and like we talked about, how fast you're spinning, the speed of that shaker. And so maintaining those forces constant is really important, and preventing the accumulation of waste in those systems is also important, which is why we talked a little bit about that media replacement strategy of potentially removing the spent media and replacing it with fresh media. Now, when those cultures get big enough, right, you'll notice that as these spheroids form, they're going to be around a certain size, usually around 150 microns. But over the next 3 or 4 days, they will eventually grow to where they get to a point where you can really easily see them in culture. But how do you know when it's time to passage, right? In 2D culture, we passage when these cells are confluent, right? When they're 75, 80% confluent. In this case, the parameter for passaging is slightly different. We passage these 3D cultures when the spheroids become of a certain size, right? When they get big enough that we have to consider those gradients that can be made across the diffusion gradients across the diameter or the radius of the spheroid, right? if they get too big, the oxygen and the nutrients won't be able to get to the core of that spheroid, which will cause it to differentiate. This typically happens when the spheroids are around 400 microns in size. Sorry, 500 microns in size. So we recommend that we passage these cultures a little bit before that, so that these spheroids are around 400 microns when we passage them. And all you have to do is basically collect your spheroids in a conical tube, spin them down so that you can replace the media with your dissociation reagent, like our StemPro Accutase, and then dissociate those spheroids back down into single cells. After which you can spin one more time, where now single cells can be resuspended in fresh media with fresh ROCK inhibitor and then seeded on to the next passage, right? So that you can do the same thing that we did when we initiated those cultures. Now, you'll see that this is the second time I kind of called out the ROCK inhibitor here. And I think it's really, really important to mention that in those 3D cultures, the addition of a ROCK inhibitor is essential for nucleation. So if I were to compare that to 2D, right? 2D cultures are we recommend adding the ROCK inhibitor when you are stressing the cells out somehow, right? You're passaging into single cells, or you know that you're bringing your cells out of cryopreservation, then we add a ROCK inhibitor there. In this case, for 3D culture, the addition of the ROCK inhibitor serves to actually help these single cells find each other and create that spheroid initially. So if you do not add that ROCK inhibitor, you don't end up creating those spheroids as well. And again, just some things to kind of consider during passaging protocols with 3D systems is the ease of the process, right? These are systems that you want to try to make them as easy and as basic as possible to minimize contamination, reduce cell loss, and eventually, if you are going to be going to those therapeutic applications, if these processes are easy enough, then they can be, scalable and amendable to automation too. And so with STEM scale, right, the idea around a suspension-based media system for PSC culture specifically is that you can, have these cells proliferate well in this media, right, in a 3D format. You can see here that for 4 representative lines, an internal, 2 IPS and 2 ES cell lines, that you can see a robust expansion of these cells over 3 passages. And we can actually work in whatever format that we want in terms of the vessel size. If I wanted to work with a little bit of spheroids, I can use a smaller well plate format. But if I wanted to work in larger formats, I can expand and scale up into either flasks all the way up to 1 liter, or even bioreactors as well. And these are what these cultures look like, right? So I flashed an image before, but these are your spheroids that have your pluripotent stem cells. Again, these are the 4 different cell lines that I showed in the previous slide, and you'll see that, as I mentioned before, they tend to it first initiate at 150 microns and then expand in size as you culture them. So as days in culture progressively increase, then so does the size of the spheroid. Where, like I mentioned, if at around 400 microns is when you want to start to passage them. Let's see, we have a couple questions. Would sept or another cocktail work better than? Great question. So we have tested the sept cocktail, and it does work a little bit better at nucleation than the Y27632. The reason why we haven't changed that just yet, which I'll talk about in just a second, is we've done longer term studies, Angel, in with the Y27632 to confirm that it doesn't introduce any mutations into the suspension-based culture system over time. And we're still waiting on that data for the culture set. So technically, yes, but we just have to confirm that long term that has no further effect on the cells. And then Kitai, good question about how easy it is to confirm the level of differentiation in those systems. So, as we talked about, right, there is very easy morphological features in 2D culture that you'll know of how these cells look like when these cultures look like when they're differentiating. On the 3D end, there definitely are also morphological features that you can keep in mind. I don't have pictures of that just yet, but I will put together a list of those images and we'll share them when available. Typically, this really nice round morphology is lost. So as these cells differentiate, you'll start to see cavitations and indentations. Almost looks like craters if you were to zoom in a little bit more. And epithelialization of that of the periphery structure into something that looks like neural ectoderm too. So we can definitely tell when these spheroids start to differentiate, and when they do, which honestly, if conditions are right, we haven't seen that before. But should this happen and should this be a very precious culture, there is a bunch of different ways that we have in recommendations on how to circumvent that and save something like that as well. Let's see, what else do we have? Seen that the borders are not very round. Fine. Okay, is it possible to transition directly from stem scale culture to embryoid? Yes, and that's what we're going to be talking about in just a second, Keita. You're right on the money. Before we differentiate these cells, right, just to confirm that these are not embryoid bodies, right? What you were looking at or what I was describing is what we're calling PSC spheroids because they do maintain their pluripotency. They are not differentiating, right? And we've done this and we've confirmed this over long-term culture, where even as high as passage 30, we see markers of pluripotency like OCT4 and nanog remain at high percentages. We've confirmed that the global transcriptomic signature of these 3D cultures also is clusters very closely to that of what is known to be for validated iPSC cell lines, but also karyotypically that we don't introduce any selective pressure on these cells through this process, so they're not accumulating any mutations at a faster rate than they would normally. So we couldn't detect any karyotypic abnormalities after 30 passages. And we also even did some NGS work where we're doing some sequencing of proto-oncogenes to make sure that these cells are not that these genes are not mutated as well. All really to say that we can culture them as and keep them as stem cells in this 3D format. Now, the really cool part is when you get to differentiate them. Now, I'm not going to steal the thunder because that's for a subsequent lecture, but we can take those spheroids that you see here in panel A, which are stem scale spheroids, in their pluripotent state. And then as we can, remove the media and replace the components in the media or what these cells are basically exposed to, we can start to differentiate them into different cell types and eventually organoids, or even EBs if we wanted to, right? In this particular example that I'm showing is just one case where we can differentiate these spheres into cardiac cells specifically, where in a 2-week process, we can change the media, remove those components that maintain the pluripotency of these cells, and add ones that are going to start to drive them down into a particular lineage, right? In this case, initially mesoderm, and then cardiac mesoderm, and then cardiac progenitors, and then cardiomyocytes. And so hopefully this works, but I'll see if I can do this little trick here. You see, in about 2 weeks, we can take those PSC spheroids that are in panel A and create those beating cardiomyocyte organoids that we're calling cardioids. They're really, really cool. And if I was giving this in person, I used to like to tap on the microphone to simulate a little heartbeat. Because the first time I saw that, I was in complete awe. These were little 3D structures that self-organize and beat in less than 2 weeks. They're like mini little mini hearts, which was so exciting to see, right? Really, really cool. They express, several different markers of cardiomyocytes like alpha-zactraminic actinin and troponin. They even were doing a little bit more characterization on them, and they tend to specify a layer of endothelial cells, which means that they're trying to vascularize at a cellular level, which is super cool. But you'll hear more about all of this stuff and how differentiation part in 2D and in 3D in subsequent lectures too. And so with that, just to kind of as a parting way, again, don't forget about some of the resources that we're going to be sharing. We do have a bunch of different e-learning modules around thawing pluripotent stem cells and culturing them in 2D and in 3D. You saw some of the how-to videos, but I've put the links in there as well. And I've kept in the link to Shinya's Nobel acceptance speech because it kind of he talks a little bit about culturing cells during that too as well, which I thought was pretty relevant. I know this was a lot, and I hope again we're not boring you to death with all this information, but I really want you to get a really good understanding of how much this field has come in the past 25 to 30 years. We really went from hardly being able to culture these cells in a very reproducible, consistent manner to methods and protocols on how we can make mini hearts in 3D settings in 2 weeks, right? Just crazy cool stuff. But I'm happy to take any questions. See if we have any questions in the chat as well. But please feel free to speak up. Let's see. Do we have any questions in the chat? Is it possible to transition directly from stem cell to culture? Oh, we already answered that. Do we know the spheres are hollow or compact cell? They're not hollow. It's a good question. It's a bunch of cells all through around because we can serially section and look at them, look at the Z stacks where we see cells just across from one end all the way to the other if you were to look through. Good question. Oh, and I
Omar,
see Tom already has it.
where does Thermo Fisher find the actors for these videos?
I was hoping you wouldn't call it out. Some of you might have recognized either Tom or myself in some of those videos. So um, uh, if you guys need somebody to film a particular how-to video or something in the lab, Tom and I are definitely ready. Our fees are quite low. It's probably just a couple of drinks or something like that.
You get paid?
Exactly. No, but seriously, in terms of resources, right? This is something that Tom and I do on the regular. So, aside from these lectures here, please, once you're in the lab or once you're working, and even if you have particular questions or want to come back and do more, a little bit more, like discuss something a little bit more in detail, let us know. We're happy to help for sure.
Hi, Omar. I have one question about the
Yeah.
differentiation of steroid that you have shown.
Mm-hmm.
For this differentiation, have you used the cardiomyocyte differentiation kit that you have, medium A and B, is the same kit that you have used?
So we have used that kit, and you are able to derive cardiomyocytes from it. But the protocol that I was showing there is actually a modified protocol that uses only small molecules, small molecule inhibitors. So a GS3K inhibitor to inhibit Wnt signaling, or sorry, activate Wnt signaling initially, and then a Wnt inhibitor to block all Wnt signaling. And then we use RPMI and B27 for that particular protocol. But we have seen relative success with both, Naresh.
Yeah, okay. Because I do have that media that I'm using for my 2D culture. So just I was wondering whether I can use that one for organoid differentiation.
Yes, no, you can. And there is a couple of considerations when it comes to the 3D differentiation side and organoids that we're going to talk about again in subsequent lectures as well. But on the 2D side, we always talk about seeding densities and starting confluences with differentiation protocols. On the 3D side, we also talk about the size of the spheroid or the EB before you even start your differentiation, right? So these are very important considerations that we're going to mention in subsequent lectures. In this particular case, Naresh, for the cardiomyocytes, what we've seen is that they prefer a larger starting spheroid. So around 300 microns or so tends to be better for differentiation with our protocol into mesoderm and eventually cardiomyocytes. Yeah, but we'll discuss this
Yeah.
in the next several calls.
Yeah. Okay, great. Thank you.
Of course. Can we switch culture medias between 3D and 2D culture? Um, you definitely can go from 2D to 3D quite readily. We have also, for a short period of time, gone from 3D back down into 2D. I have to just confirm that we have taken those cultures out long term as well. When it comes to the switching of the cultures, yes, but the use of the different medias perhaps may not necessarily be the best choice. So, because I have gotten this question before, of whether, for example, they can use StemFlex for 3D culture or StemScale for 2D culture. And I would say the way the medias have been designed is specifically for their intended 2D and 3D applications. So, just to call that out as probably the recommendation there is to use the appropriate media depending on whether you're in 2D or 3D.
Hey,
Any
Omar,
other than
question.
yes, please, Aisha.
So we have recently started using the CTS stem scale
Hmm.
medium. And when you are transitioning from a 2D format in StemFlex to the CTS stem scale, the spheroids, I mean, the growth is usually slow, like for the first passage or the
Hmm.
second passage, and then they pick up and you get the 8 to 10 fold increase in the cellines, right? So for the
Correct.
first and second passage, a lot of the times we are currently doing it in 6-well plate. So
Hmm.
we have to seed, say, 500 microliters of the cell suspension in order to get to the 0.15 to 0.2 million per ml density. So,
Okay.
because when we have a lot of plates, I always wonder that it takes time to remove the 500 microliters of medium and add the cell suspension. So I understand the media volume might, what I want to understand is, does the medium volume impact the size of the cells, size of the spheroids? For example, for a 2 ml of recommended volume for a 6-well plate, if it goes up to 2.5, could that impact the size of the spheroid?
Very good question. So yes, the short answer is yes, Aisha. The in terms of the this hold on, let me kind of take one step back to explain this a little bit better. There are two main factors that will affect the size of your spheroids as they form, right? One we already kind of briefly discussed, which is the speed, right? At the speed at which you rotate. But the volume of the media in your culture vessel tends to also affect that, right? So if you think about it, it's with the same amount of energy, if you're at an RPM of 70, for example, moving 2 ml of media is going to be much easier than moving 2 liters of media, right? So in that case, the volume, the energy in the system, right? The forces that we want to keep constant in the system depend on your rotational speed and your fill volume. So in your case, for example, having a 2.5 ml fill volume in your 6-well plate might be a little bit higher than what you'd want, because then the speed isn't optimized for that particular volume, right? You may want to crank up the speed a little bit more. Alternatively, reducing the volume back down to 2 ml and keeping the same recommended speed would be what I would typically recommend, because manipulation of speed is much more easily tunable than manipulation of volume in this case. Does that make sense?
Yes. Yeah. Thank you.
Yeah. Yeah, but basically, short answer is technically you can increase the volume up to 2.5, but you would have to proportionally increase the speed, and that can be a little bit trickier. So I always recommend just to keep the volume at the recommended, and then play with your speeds if you need to. Yes, Naresh, sorry, I did see your email. I was just traveling last week, but this will go out. It's in my inbox. It will go out to you by the end of today.
Oh, yeah, okay, thank you.
Excellent. So with that, we'll call it a Tom, Kita, anything else on your end?
Nothing much. Next week is we are going to talk about the gene editing. A number of people really interested in this section. You can join review by YouTube channel, but directly attending class with chatting communication, that is actually more fun. So therefore, please join the regular lecture. Of course, you can join as a YouTube channel.
Nothing else on my end. Great job, Omar. Thanks.
Perfect. Thanks. And I know we are getting into the weeds here, right? We're throwing a lot of information at you. At some point, trust me, even I get a little bit of a uh, uh, I would say messed up completely, but like I say the wrong terms or misuse words sometimes. So um, uh, just as a halfway check-in, because I know we're halfway through the course right now, or around lecture 4. Uh, how are folks feeling? Are you getting a better concept or better understanding of the different concepts here that we're talking about? Is there anything that you'd like to see expanded upon? Anything that isn't very, very clear? Let us know. We can go over any and all of this honestly too as well, either on a one-on-one, or if need be, we can add more lectures to the course so that we can have, or office hours for folks to join if need be. So let us know. We're happy to help. I'll take your silence so far as everything is hunky-dory, and you guys are all now ready to do 3D cultures of cardiomyocytes beating cardiomyocytes. No, it's soon enough. No, definitely. Perfect. With that, I'll say thank you again for your time. And if there's anything, please let me, Tom, or anybody on the UCLA side know, and we're happy to help as well. Perfect. Have a good one, everybody. And
Thank
happy
you.
Thank you very
Monday.
much. Great job.
Thank you, Omar. Thank you. Bye.
Great job.